Biotechnology is a fascinating field that merges biology with technology, allowing us to manipulate living organisms or their systems for practical purposes. At its core, biotechnology relies on a set of principles and processes that have revolutionized medicine, agriculture, and industry. From the amplification of genes to the creation of genetically modified organisms, the journey begins with understanding the tools and techniques that make these advancements possible. In this video, we'll explore the foundational concepts of biotechnology, including the role of restriction enzymes, cloning vectors, and the significance of palindromic sequences. As we move forward, you'll see how these principles are applied in real-world scenarios, setting the stage for innovations that impact our daily lives. At the heart of recombinant DNA technology are specialized tools and techniques. Restriction enzymes, often called molecular scissors, recognize specific palindromic sequences in DNA and cut them at precise locations. For example, EcoRI recognizes the sequence GAATTC and cuts between G and A, producing sticky ends that are crucial for joining DNA fragments. Plasmids, which are circular DNA molecules found in bacteria, serve as cloning vectors to carry foreign genes into host cells. Selectable markers like antibiotic resistance genes or beta-galactosidase help identify successful recombinants. Techniques such as microinjection, gene gun biolistics, and heat shock are used to transfer recombinant DNA into host cells. Each method has its own advantages, depending on whether the target is a plant or animal cell. These tools and techniques form the backbone of genetic engineering, enabling scientists to create new genetic combinations for research and practical applications. Once the gene of interest is isolated, amplification is essential. The polymerase chain reaction (PCR) is a powerful technique that can amplify a specific DNA segment up to 1 billion times, using thermostable DNA polymerase like Taq polymerase from Thermus aquaticus. PCR involves cycles of denaturation, annealing, and extension, allowing rapid multiplication of DNA. After amplification, gel electrophoresis is used to separate DNA fragments based on size. DNA, being negatively charged, moves towards the anode through an agarose gel matrix, with smaller fragments traveling faster. This separation is vital for analyzing genetic material, conducting DNA fingerprinting, and preparing samples for further genetic engineering. The combination of PCR and gel electrophoresis has transformed biotechnology, making gene analysis and manipulation more efficient and precise. Scaling up from laboratory experiments to industrial production requires bioreactors. These specialized vessels provide optimal conditions for the growth of genetically modified organisms and the production of desired products such as proteins or enzymes. Stirred tank bioreactors are commonly used, offering advantages like efficient mixing, oxygen transfer, and temperature control. Operation of a bioreactor involves careful monitoring of parameters to maximize yield and maintain product quality. After the biosynthetic stage, downstream processing is necessary to purify the product before it can be used commercially. Bioreactors have enabled the mass production of pharmaceuticals, biofuels, and other valuable biotechnological products, making them indispensable in modern biotechnology. In summary, biotechnology principles and processes have transformed the way we approach challenges in health, agriculture, and industry. From the precise cutting and joining of DNA to the amplification and large-scale production of valuable products, each step relies on a deep understanding of molecular biology and innovative technology. Tools and techniques discussed, restriction enzymes, cloning vectors, PCR, gel electrophoresis, and bioreactors, are the foundation of modern genetic engineering. As research continues, biotechnology promises even greater advancements, offering solutions to some of the world's most pressing problems. By mastering these principles, scientists are paving the way for a future where biology and technology work hand in hand for the betterment of society.